Great, so it's my pleasure to welcome George Church, who's going to give us a talk today on reading and writing Omics in situ. And so here we go.
I'm looking forward to the question and answer part of this. Feel free. This is my conflict of interest slide, as many of you know. And if it's going too fast for you, it's online. This is the kind of topics I think are relevant to this group and increasingly in biology is how we evaluate our ability to perturb systems, which I would call writing, by more intense levels of reading, meaning at subcellular resolution, but whole body scale, possibly whole ecosystem. So we have this huge range of our ability to read and write. I'll give one example. When people talk about writing genomes these days, they tend to talk about editing. I'm going to talk about something that's quite a bit broader than that, which we call Genome Project Write, which we think is the logical successor to both Genome Project Read, which we just finished, and also for editing. And then this panomics or omniomics, to which we add the word in situ, and we can apply this to all your major food groups, DNA, RNA, proteins, and even metabolites, which I probably won't talk about unless you insist. And then when you can do omniomics, then you have this opportunity to integrate all sorts of data types, and I think it's incredibly important. That problem of integrating as not just leading to understanding, but leading to a closed loop where you can write systems, which writing in this case can be either testing hypotheses or developing therapeutics. And then finally, variants of unknown significance. So that's what we're talking about. This is, in various forms, this kind of plot is seen again and again at meetings like this, usually just the reading line, which is the red line, which is for reading human genomes. But the blue line is for writing DNA at both of them in units of nucleotides per dollar. And many people are shocked that the writing curve is so close. And they almost take for granted at this point that the slope changed from the Moore's law curve, which we had for decades, and then suddenly changed slope for both of them. So, and they're both sort of in the $1,000 range for a human genome, a high-quality human genome that's interpreted, and sort of in the couple, $2,000 or $3,000 for writing 200 mers. Sort of the reading and writing raw reads are about the same length. In any case, the slope changed, I would argue, because of miniaturization and multiplexing, which is a form of miniaturization where you mix together compounds or nucleic acids. And that happened in 2003, 2004 for both reading and writing DNA on chips, essentially, on solid surfaces. Now, you might say, well, I understand why we might want to read the genomes of 7 billion people and maybe their microbiome on a regular basis, but why would we want to be able to have large-scale writing? And I'll just give you a few examples here where we've done gene assembly and now genome assembly from chips, not just ordinary organic chemistry. We can make short hairpin libraries, a whole variety of different libraries, guide RNA libraries, cis-regulatory. We can barcode everything almost, I mean, I'm sure you're all familiar with all the barcoding strategies, some of which date back to the 80s. And then almost every biological system, you can turn it into a library and you can turn it into a screen or a selection. Not on here is other work that we're doing, some of which in collaboration, in close coordination with Microsoft on encoding digital information into DNA and analog information for that matter. But let's talk about editing DNA and how limited it is. I should be saying how great CRISPR is, having been somewhat involved, co-conspirator in turning it into a technology. But I've been ever since before we invented it in 2013, I've been advocating going beyond it. And so this is one of many Beyond CRISPR slides. It's the only one you're going to see. And there's off-target effects that we talk about a lot, and there are various solutions. The biggest one being just to look at the genome computationally before you design any guide RNA. I'm assuming maybe a little audience participation. How many people here have never heard of CRISPR? Whoa. Okay. So, well, you'll hear a little bit about it, but I'm mostly saying that forget about it. It's already out of date. It's 4 years old. But there are alternatives to this thing that will not be named, which are in which you can replace DNA in a genome. This is basically a way of editing DNA. You can, when you use CRISPR or a variety of other editing methods, what you do is you cut and then you hope for the best, where if you have a repair oligo that is what you want, doesn't catch up to the cell will make a mess, and that mess is called non-homologous end-joining. Some people glibly call it genome editing. I call it genome vandalism. True editing is reserved for when the cut and paste actually works and you get what you want, or homologous recombination. Anyway, there's two other enzyme categories which are much better at not making a mess. They don't have a side reaction which is non-homologous end-joining because they don't make a cut and then hope for the best. They actually have a coordinated activity. And these are recombinases and integrases. And just to demonstrate how powerful they are, we get really excited about editing 1 or 2 genes at a time, 1 or 2 base pairs at a time. Here's an example where we are editing 4 million base pairs at a time. So that really no longer call it editing, we call it writing, although it is inspired by natural systems. And I think it's important in each of these, computational biology / technology projects to say why we're doing it. And the why here is that we can extend the vocabulary, the alphabet of amino acids in proteins by many different amino acids. We can do genetic and metabolic isolation, which is biocontainment. And most importantly, profoundly, we can get not just multi-virus resistance, but in principle resistance to all viruses, even viruses we've never studied before. And you might say, well, how could you do that? You don't know what their receptors are. You don't know what their mechanism is. The answer is that all viruses, and I think it's pretty safe to extrapolate, have a genetic code requirement. And most of the codes in the world are, in all organisms, have some things in common. In particular, they use all 64 of the 4 to the 3rd triplet codons. And so we made an organism that has only 63 codons in use. And that one was resistant to a surprising number of viruses. And now we're almost done making a genome which has only 57 codons in use. There's no other organisms naturally or synthetically that we know of that has less than 64, the full set. And so some of this work has been reported in Science and Nature, but we have this most recent paper where this is already out of date, even though it's only a few weeks old. We're much closer to complete synthesis of this 4 million base pairs. And I should mention this was done entirely without CRISPR, which for most of you is not shocking because you hadn't heard of CRISPR. But anyway, it was done with these 2 other enzyme categories. And this was out of the 4 million base pair genome, there were 148,955 changes made. That's quite a few edits. But back to the topic of reading omniomics and integrating data over many different categories. And I was also a co-conspirator in I think, a more impactful project, which is now called the Brain Initiative. And one of the things we dealt with was some of the physical limitations. In other words, rather than thinking it from what current technology limitations we have, what are the principles of physics that we're going to eventually run up against boundaries? And they're really not as limiting for brain analysis as one might think. And so we categorized sort of our wish list of 5 food groups here, which is the activity of the brain, behavior of the animal in response to what they are seeing, sensing more generally, and then their motor activity. The connection of the brain, so in other words, each neuron makes maybe 1,000 connections, and that synapse level information is very fine-tuned, very small, on the order of 100 nanometers, sometimes less. Developmental lineage, how we went from one fertilized cell to 10 to the 12th or more cells. In other words, at each fork, there's exactly 2 daughter cells, and then they have two, some of them die, but and move around. But anyway, that's the developmental lineage. We want to be able to track that. And finally, the expression. This is something I'm sure you're all familiar with. DNA, RNA, protein, central dogma. That's the expression. And we do this by a method that I'll just briefly tell you called fluorescent in situ sequencing, and we think that almost all of these can be integrated by doing this microscopy method on all of them at once. And so I'm going to lead you through them one at a time, starting with activity. Behavior is a given. You have that. That's kind of the external meta laboratory setting. What inputs you're given to the animal or the human, and what outputs they're producing. But the activity that's going on within their brain is typically monitored by calcium imaging, where you have a fluorescent calcium response to the but an alternative. The problem with the imaging of calcium in a large brain is that it's hard to get good optics to go deep, and it's also hard to embed enough optical fibers so that you can detect, even if you inject them invasively throughout the brain, it's hard to get monitoring of all the neurons simultaneously, and it is a system that merits that. So we have 2 approaches to measuring activity, just to stimulate the way we're thinking about this. And this is related to our interest in recording information into DNA. So DNA is now like a tape drive or a flash drive, but it's much smaller, and it is much lower energy consumption. And so that's what makes it appropriate. It means that we can now put this tape recorder essentially in every cell of the brain, every cell of the body, every cell, possibly subcellularly, because there are thousands of synapses per cell. Anyway, here's one example where we're literally monitoring calcium, and in the next example I'll show you we're monitoring nucleic acids. So this is, here we noted that DNA polymerases in general have a sensitivity to divalent cations, and so here we could use mixtures of magnesium, manganese, and calcium to get a calcium response, and where the normal base pairs A with T and G with C you can add a mismatch here with a GT mismatch, and that and all the mismatches can be increased by changing the divalent cation in an interesting physiological range here that you might find in a neuron. And this is Brad Zamp did this as a postdoc. And I should mention DNA polymerase rates are up to 1,000 hertz, 1,000 events per second. Here's a much slower process, and it's more appropriately matched to the process we're trying to measure, which is nucleic acid variation in a cell. Normally, we'd be most interested in single-stranded messenger RNA. In this case, we use single-stranded DNA as a model system. And here, we're using another part of CRISPR that you haven't, that even those of you who've heard of CRISPR as an editing tool probably haven't focused on this, which is the mechanism by which you memorize invading phages. You take a little piece of their genome, and if you survive the first viral attack, you'll remember forever that that's your enemy and you're going to attack it. Anyway, we took this system. I'm trying to emphasize the data aspects of this rather than the molecular biology aspects of this, so I apologize for being so terse about the molecular biology. But anyway, the system is Cas1 and 2 rather than the famous Cas9. And we made a modified Cas1 and 2, which has a different consensus sequence that it recognizes. an invading molecule. And so then we can make these two systems responsive to different small molecules. In this case, one's responsive to lactose molecules and the other one to tetracycline-like molecules. So you can program it from the outside to go up and down into what kinds of nucleic acids it's encoding. And so you can see here, these are a time series of 3 different time points. This is the very basic, most minimal proof of concept experiment, is we can get the two systems to go in opposite directions, be anti-correlated. And we can basically encode anything we want. There's a paper right after this one, which is in review, where we've encoded videos of a horse running, a classic video, has been coded in using this CRISPR-Cas1-2 system. Here's another cute trick you can do with CRISPR. Now I will finally explain it, what CRISPR is. This is the famous one. This is the Cas9. And this is the textbook version of it, where you have this little arrow means transcription initiation. And the blue and the red are all part of one RNA. That's part of the technological leap, was to fuse the spacer to the scaffold. Now there's one fused RNA. Anyway, the simplest thing is that you transcribe this guide RNA. It binds to the purple protein. And then as a complex, they will bind, making a triplex, a 3-stranded interaction with the genome at a target. And this target here is indicated by homology to the blue spacer region and a yellow region, which is the protein recognizes, called the PAM. Anyway, this is very simple. You transcribe the guide RNA, it binds the protein, they bind to the genome, and you cut. You make a double-strand break. That's what it does in vivo, evolutionarily, and that's what we've adapted as a technology. But now we've done something very perverse, and again, this won't be shocking to you, those of you who haven't worried about this. But instead of targeting the normal target, we target the guide RNA gene itself. So the guide RNA now, normally, when it hits the target, that target gets damaged and is no longer a target. In this case, it hits itself, and so then it becomes not a target for the original guide RNA, but the new guide RNA is made by this transcription start, and it now becomes a target for itself again. So this kind of a self-modifying code. Probably for the coders in the room, this seems like very bad coding principles to modify the code while the code is running. But in any case, this is good for generating diversity, and our point is to generate very tight, this is another trick of encoding information as you go. So as each cell divides, it's accumulating mutants in each of these self-modifying barcodes, so that the daughter cells have a different barcode, and then their daughters have a different barcode. And you can track the lineage. You can reconstruct the phylogenetic tree, as I'll show you in the next slide. And we did this along with 2 of our previous collaborators from the Shinduri lab and Tim Liu's lab. So this is the idea of reconstructing a tree, which I don't have to tell this audience. But here, we're not reconstructing a phylogenetic tree, but a lineage tree of the cells. So here's the original. This could be either the original fertilized egg, or it could be any single cell along a developmental lineage. It divides into 2 cells, almost always exactly 2 cells. And then they accumulate some mutants in the self-modifying or homing CRISPR barcode, and it keeps these final cells in the tree are clearly more related to each other than they are to the other 2, and so forth. And so you can reconstruct the tree. Now, to read that out, we want something which has the exquisite resolution of single cells. In fact, subcellular resolution, because, like I said, a neuron will branch out into 1,000 different connections, and you want to be able to tell those apart. It is not well approximated by a single sphere adjacent to other single spheres. It is much more complicated than that in a way that's extremely important. So we call this method in situ sequencing, or FISSEQ. It is now an instrument that's available and a service that's available for people who don't want to buy the instrument. And it's related to a method called Oligopaints that I'll mention in a moment from the Ting Wu's lab. And this is, for those of you who do sequence analysis, this is multidimensional separation of the 4 colors, A, C, G, and T, and it's very clean. And this is done in situ. So it's very similar to next-generation sequencing, but we don't throw away the 3D information. Here are 2 cells. Each dot is a single RNA molecule that's been lightly amplified so it doesn't spread its signal out. And you can see they're very clean. There should be 4 different colors. There should not be mixtures of colors, and that's what we observe. They're very, and and so each of those dots, that was a snapshot of one scenario, just like this one. Each of these dots goes through multiple cycles, and so what you'll see is, just a moment here. Each dot changes color as you go through cycles of sequencing. This is just like next-gen sequencing, except it's in situ. Now we're going to change the way what we're displaying. We're now showing the time sequence for each dot, each single RNA molecule in the cell, and you can see each one of them has a very colorful barcode telling you exactly which molecule that is, which you then do computational search on it. Now this is to show that you can detect alternative splicing. These are a histogram of reads along one of the thousands of messenger RNAs that we detect in situ. This is a particularly long messenger RNA, 8,000 base pairs long. And here is missing data. So it's not just that it's weakly represented, it's absent. It happens to correspond to an exon. So missing that would be unfortunate, but it turns out it's alternative splicing. So you can detect alternative splicing in situ and point mutations, since it's a sequencing method, including allelic changes as well as RNA editing. Now that was RNA, this is DNA, and this is done at super resolution. So this shows kind of what we will call conventional super resolution microscopy, although it's very unconventional oligonucleotide paints. It was developed in Ting Wu's lab at Harvard. This is showing conventional resolution, this is super resolution, this is a blow-up of that super resolution section. And you're getting on the order of 20 nanometers, or sort of the size of a nucleosome in this method. And you can trace the chain from telomere to telomere, or at least over large chromosomal regions. So this could solve haplotype phasing problems, as well as you get the 3-dimensional structure of the nucleus as a bonus. Now the fourth method that, again, illustrates a new polymer type, which is proteins, as well as a new microscopy type, which is expansion microscopy. So rather than trying to make super resolution, where the physics is you query a single point over and over thousands of times to get the centroid better, that's slow. In this case, you expand the sample literally, isotropically in all dimensions, and now you can get high resolution with conventional, meaning very fast microscopy. And in this case, the purple proteins are detected by the Y-shaped gray antibodies, which are now coupled to a nucleic acid, reducing a protein problem to a nucleic acid problem, which we've already solved, you know, as mathematicians would tell you. And this can now be, you can now read out any protein for which you can make a labeled antibody. This has been validated with comparison to other super resolution methods, and we can do this at large scale and at synapse level. Here we can tell the pre and postsynaptic proteins, so we can know which, when you have a synapse, which way the information is going. Very important for the brain. Here is pan-omics or omni-omics, DNA, RNA, protein. Here is an example of expansion microscopy. I don't need to go through it. All of the samples that we use come from this project, and so if you're interested in shareable data, this is the most shareable data in the world. It's called the Personal Genome Project. It's international in 6 different locations. Approval for re-identification and commercial use, and hence this is the source of the world standard now for NIST and FDA for genomics and also cell biology that we've been doing. And so one application of this is variants of unknown significance. We're getting more and more of these from the diagnostic laboratories, and we'd like to be able to add a pipeline, which is high throughput, to get it from correlation to causality. And some of these have N-of-1 studies where you don't even have correlation. You can do it with animal models, but the emerging possibility is to do it with with isogenic pairs of cells. So basically what you do is your computer human interface will generate predictions, possibly hundreds of predictions of what there might be 3 million differences between you and me, but there might be a small number that explain hypothetically a particular disease. In this case, a cardiomyopathy that Bill Poo brought to our attention and collaborated with us on and trying to determine the causative allele. Here is one of the causative alleles, possible, putative, potential causative allele, where a G is missing on the X chromosome, throwing the TAS protein out of frame. That seems like that's a good candidate, but it's not proven until you do this experiment. So the experiment is to take these PGP1 stem cells with the CRISPR-Cas9 to use it a repair oligo, so not the vandalism, but the actual real editing. And then when you do repair, then you get just one change in the genome. So now, unlike this baby and me, who differ by 3 million base pairs, we now have so I'm PGP1, my stem cells with and without that guanine. And you can say, well, there's all this off-target editing. How do you know that you really have only changed one place in the genome? And the answer is, and it's very illustrative of how far we've come on this exponential journey, that now postdocs and graduate students think nothing of sequencing a whole human genome, 6 billion base pairs, just to check that one base pair they changed is what they wanted. So you do clonal stem cells where all the cells are basically the same. You take a few cells out and you sequence them, and this is now routine. And so that's what we did in this case. And it also illustrated that you need to know your genome, because knowing a generic human genome is not sufficient. You need to know the genome that you're actually editing, because a single SNP can create a new off-target, which we discovered in that study as well. Luhan Yang studied that. So anyway, here is, you now need to be able to test that. You've made stem cells that differ by one base pair, but you actually want, in this case a cardiomyopathy, you want the correct organ to test it in. And it's hard to engineer human babies and harvest their heart for science. So we didn't do that. We made organoids in the lab, in this case something approximating a cardiac tissue in a heart. And this was off by one. So these differ. This is a control that was mock modification. This is modified at 1 G out of the whole genome, and you can see differences in morphology, physiology, contraction, biochemistry, and so forth. So this proves that one base pair was the cause. Now we want to generalize this. We want to be able to make any kind of organ, any kind of cell type. And so in order to do this, we wanted to generalize the great success of groups like Shinya Yamanaka, who used 4 transcription factors to reprogram fibroblasts from the skin into stem cells. We want to be able to reprogram anything into anything. For that, we needed to get a library of all the human transcription factors, which was surprisingly far away when Alex started this a couple years ago. But now we have an example of every gene that's been annotated as a human transcription factor, and in some cases, multiple splice isoforms for those. And then we've done whole libraries of these for various assays like differentiation and dedifferentiation. And I haven't got time to go through all that, but we can make any neuron you want in really, really rapidly and efficiently. So here's protocols used to be 4% yield in 4 months. Now we have 4 days and 98% yield, and we're just getting better and better at this. And then just one parting comment about this is that there are real cerebral organoids. These are brain in a bottle sort of things, but they're limited to less than half a millimeter because they don't have vasculature. We now have ways of generating vasculature. I can talk to anybody that's interested in that. So I just want to end by thanking my many collaborators I mentioned along the way, plus this team that has a Center for Genomically Engineered Organs, and this IARPA-funded Brain Initiative project where we're spending $100 million to analyze a cubic millimeter of visual cortex. Cubic millimeter. It's one of the we're talking about something in the multi-petabyte range, and IARPA isn't doing this for the biomedical insights, which I think will be many. They'll be considered collateral damage by IARPA. What they're really interested in is deep machine learning algorithms that will come out from knowing exactly how a rodent processes visual information by studying activity and connections in that cubic millimeter. So finished with 4 seconds to spare. Lots of time for questions. Thank you.
Questions?
You started this great talk with talking about recording, and I was wondering if you could explain a little bit. I think you went a little bit too fast for me, at least, about
probably 99%. Yeah, yeah.
How the data is how the time space correlation of the recording actually happens.
Okay. Right. So this is a really long topic, but I'm going to give you what I think you want. Which is space is recorded because the recording device is in a particular cell and you leave it there. So that's the whole idea behind in situ sequencing is rather than breaking things apart where we lose the 3D coordinates, we keep it exactly where it is, where we have resolution down to 20, 30 nanometers if we want it. So that's the XYZ. The time component has been difficult because if you're trying to monitor a dynamic activity like calcium or RNA or protein, you kill a thing and you got a snapshot. You got beautiful 3D coordinates now, but you've lost time. So you want to encode the time in a compact recording device. Well, cramming in even the smallest electronic coding device starts to take up serious space in something as delicate as the brain. So we put synthetic biology recording devices into cells, embedded in the cells. And I gave you 2 examples, or actually I gave you 3 examples. One is for calcium, which we did in vitro, full disclosure. Then for the nucleic acids, which was a toy system with single-strand nucleic acids, which we used in bacterial cells. And then finally, we did developmental lineage encoding the time series of how the cells divide and differentiate into new cell types. That was done in mammalian cells. That was done in mouse cells. But all of them, in principle, can be done in a variety of in each of those ways. And the way you encode the time is you'll there are 3 different ways we encoded the time. But in each case, it results in a change in the DNA sequence. And so later, when you do the in situ sequencing, you can reconstruct from the digital information along the DNA polymer, you can reconstruct the time series, which is in exactly that same order. That's the basic idea. And we have other experiments which are completely non-biological where we encode it by chemical synthesis.
Any other questions? I'm going to ask a question. So in all the kinds of experiments you're doing, is there a particular area you think would be ripe for maybe people in this audience who are computationally oriented, machine learning oriented, that you think would be?
Well, every one of these. I mean,
okay. Could you elaborate on a particular one that you think is?
Well, so I think for the DNA encoding, you've got to deal with error models. You've got to integrate. I mean, integration was a theme here. I wish I could have gone through all the. I mean, every one of these has a computational component. But if I'd gone through the computational component, we would have made it through half of one of these topics rather than give you, hopefully, stimulate you to think about what the computing is behind it, our primitive computing we have behind it right now. But for the developmental biology, you want to be able to do the reconstruction of phylogenetic trees, which is not a foreign topic, but it's going to be in a new context here. And the list goes on. You know, for in situ sequencing, we've got sequencing tools, computational sequencing tools, and we've got computational tools for image analysis, but merging those in a meaningful way and merging them with everything we know about developmental biology is a terrific computational task. I mean, I think guys should really be salivating over each of these topics, and that was at least my intention.
Great.
I have a question for you while we're waiting, if there's another question. How many people here have their full genome sequenced, or even a major portion of it? That's better than most audiences these days. Most audiences is closer to 0. So anyway, I think you all should get your genome sequenced. It should be part of your employment package. Yeah, I'm serious. Yeah.
Do you offer it to your employees?
Yeah, you got it. We're even worse than that. We have a project where we give away genome sequences, and my staff is excluded from that. So I'm even worse than a benevolent company. But there are ways that people get sequenced. So in the Personal Genome Project, essentially all the genomes, we've done hundreds of genomes for free. So I guess that compensates. They're virtual. Yes.
You
Yeah,
can give them as Christmas presents,
Yeah.
right?
It's a good idea. Yeah.
Great.
Okay.
I think we're out of time, and so we're going to move on to the next speaker, but let's thank George Church for a great talk.